Did you know? Available with dubbing in other languages. Activate it in the audio track settings. How could science bring back the dire wolf, extinct 10,000 years ago? For 3 seconds, imagine this. A huge wolf ancestor, a top predator of the Ice Age, stepping out of the mists of time to walk in our world again. Is this a fever dream of science fiction, or a real possibility that modern science is starting to consider? Well, this is where myth meets science. The dire wolf, whose scientific name is Canis dierus, was one of the most formidable carnivores of the Pleistocene megafauna. It roamed the Americas for hundreds of thousands of years, but mysteriously disappeared about 10,000 years ago, along with many other large Ice Age beasts. For a long time, based on the evidence from its skeletons, we assumed it was simply a bigger, stronger version of the grey wolf we know today. It looked like a giant grey wolf, weighing up to 25% more. But here's the twist that changed everything. A groundbreaking genetic study published in the journal Nature in 2021 analyzed ancient DNA extracted from dire wolf fossils. The results were shocking. Genetics show it was a completely separate lineage. In fact, it was so different that its lineage split from that of grey wolves almost 6 million years ago. This means that, despite their physical similarities, it likely couldn't have bred with modern wolves. It wasn't a close cousin, but a very, very distant relative. That genetic detail changes everything. It means we can't just upgrade a grey wolf. Bringing back the dire wolf is a much more complex challenge. So, if science were to take on this monumental challenge, how would bringing it back actually work? Let's explore the options. Option 1, the most straightforward, using ancient DNA. We have a lot of fossils from places like the La Brea tar pits in California, which have preserved bones exceptionally well. From these fossils, scientists can extract DNA fragments. But here's the problem. Time is DNA's mortal enemy. The molecule has a half-life of about 521 years. After 10,000 years, what's left isn't a complete strand, but billions of tiny, broken, chemically damaged fragments. Using powerful computers, scientists can sometimes piece together a genome from these fragments, like putting together a million-piece puzzle without the picture on the box and with many missing pieces. This reconstructed genome is incredibly good for getting information. We can learn about its biology, its evolution, and the reasons for its extinction. But it's very bad for perfect Jurassic Park-style cloning. For that, we'd need a complete, intact genome, something that simply doesn't survive that long. This brings us to option 2, the more realistic approach, the substitution or proxy approach. The idea is to take the genome of the most suitable living species, which despite the evolutionary distance is still the grey wolf, and use gene editing tools like CRISPR-Cas9. With this technology, scientists could, in theory, make thousands of precise edits to change the grey wolf's genetic switches and bring them closer to those of the dire wolf. This would involve modifying the genes that control development to achieve a larger, more robust build. Genes responsible for bone structure would be edited to create stronger, bone-crushing jaws, a key characteristic of the dire wolf. And crucially, different immune genes would be adjusted to reflect its adaptation to ancient American pathogens. However, each edit carries a risk of off-target effects, unintended changes elsewhere in the genome that could have unpredictable consequences for the animal's health. Once you have an edited cell nucleus, it would be inserted into a grey wolf egg cell that has had its own nucleus removed. This reconstructed embryo would be grown in the lab and then implanted into a surrogate mother, likely a grey wolf. It's important to understand that the result would not be a pure dire wolf. It would be a genetically modified grey wolf designed to look and hopefully behave like one. You don't get an identical copy, you get a functional substitute, an ecological stand-in designed to fill the niche the dire wolf left empty. A third option, even more futuristic, is cloning from stem cells. This technique would involve taking cells from a living animal, like a skin cell, and reprogramming them to become induced pluripotent stem cells. These stem cells could then be guided to become gametes, that is, sperm and egg cells. Then, in vitro fertilization would be performed to create an embryo that would be implanted into a surrogate mother. But here's the catch. This method requires living cells, or at least perfectly preserved cell nuclei. Since we don't have intact dire wolf cells, we're back to square one. We'd need to create those initial cells through intense genetic editing of gray wolf cells. So, again, we end up with substitutes. The technology is amazing, but it inevitably leads us to the tough questions, the ethical and ecological issues. First, where would this animal live? The world has changed drastically in 10,000 years. Its ancient habitats are now crisscrossed by roads, cities, and farms. Is there enough wild space for a large predator? Second, what would it hunt? The dire wolf evolved to hunt megafauna like ancient horses and bison, prey that no longer exist or are very different. Could it adapt to hunting modern deer and elk? Or would it come into direct conflict with humans by hunting livestock? And the question of disease is critical. Could this lab animal, with a reconstructed immune system, transmit new and dangerous diseases to wild canid populations like wolves and coyotes? Or conversely, would it be extremely vulnerable to modern diseases for which it has no evolutionary defense, condemning it to suffer? Any real responsible project would need strict biosecurity from the start, with state-of-the-art containment facilities. It would require detailed habitat plans, ecological modeling, and a long-term strategy that prioritizes animal welfare above all else. The question, can we do it, isn't enough. The real tests are, should we do it? And if the answer is yes, how do we do it ethically and safely? This requires the involvement not just of geneticists, but of ecologists, veterinarians, ethicists, government agencies, and the public. If such a project were to move forward, here's the likely timeline. The first phase, lasting several years, would be purely laboratory-based, mapping more dire wolf genomes to get the most complete genetic picture possible. Then, the prototyping phase would come. Scientists would test editing key traits in gray wolf cell cultures to see if the genetic changes produce the desired effects at the cellular level without causing problems. Only after years of laboratory success, and if ethical boards and regulatory agencies approve, could the next stage be attempted, small-scale pilot births in a completely contained and carefully regulated environment, with a team of veterinarians monitoring every aspect of the animal's health and welfare. And even if those first individuals are born healthy, the project's real success would mean decades of careful monitoring, studying their behavior, their health, and their impact before even considering a limited release into a protected area. So, to sum up, the myth, the fantasy, says, let's bring it back, imagining a magical resurrection. Science, on the other hand, says something much more nuanced. Perhaps, someday, we can build a responsible substitute that restores lost ecological functions, but we must do so with immense planning, caution, and ethical oversight. And now it's your turn to think like a scientist and an ethicist. Should we invest the enormous resources needed to recreate a predatory canid in the hope of healing past ecosystems, or should we focus all our efforts and funds on saving the wolves and other predators we already have and that are endangered today? Let us know your thoughts in the comments. And if these topics fascinate you, share this with a friend who loves the mysteries of the Ice Age and the future of science.